Hi there, let's kind of continue on with our blood typing content, and let's see what we can do about moving along with this. So remember that antibodies are a substance that reacts with antigens. Antigens are that protein that are attached to the red blood cells. And so, for example, if an A antigen gets an A antibody, then it's going to cause this agglutination to occur, this clumping, and that is a bad thing. So here you have the antibody. Let's say it's an A antibody. This would be an A antigen. You can see that the shape is the same. It fits, and it would cause clumping to occur. If we look at the different blood types, with type A blood, that means that you have a type A antigen. Okay, you have the A antigen on your red blood cells. And then in your plasma, you would have the B antibody. You don't have the A antibody in your plasma because then that would react with the A antigen and clumping would occur. If you have type B blood, that means you have the B antigen on your red blood cells, and you have the A antibody in your plasma. If you have type AB blood, that means you have both the A and B antigens on your red blood cells, and so if either one of these two types of antibodies are in your blood, then they would clump, they would attach to, either the A or the B, depending on which one. So that means that if you have type AB blood, you don't have either one of the two antibodies in your plasma, in your bloodstream. If you have type O blood, then you have both antibodies in your plasma, and you cannot receive any blood from someone else. You can't receive red blood cells from someone else that has A or B because you have both of these antibodies in your bloodstream, and a reaction would occur right away, and you'd have clumping occur. So that would be a bad thing. So let's take a look at a chart here that breaks everything down. So you could memorize the whole chart, but really you should be able to fill this in as needed. So whatever type blood you have, that means that you have this type of antigen. We're going to also look at the Rh factor, but it's basically the same idea. So you have type A blood, A antigen, that means you have the anti-B, right? You have the antibody B. If you have type B blood, type B antigen, you have the antibody A. So if you have type A blood, you of course can give to somebody else who has type A blood, but you can also give to somebody who has type A B blood, because all you have is that that antibody B, and they can receive that as well. You can get blood from type O or type A. So type O is essentially going to be a universal donor. Type O negative specifically is universal, and so that's something we'll get to in a moment here. And in fact, let's go ahead and look at the type AB blood. So if you have type AB blood, that means you have both antigens. Okay, you have both the antigens. And the antibodies in your bloodstream, as we saw from the little graphic earlier, you would have neither the A nor the B antibodies in your bloodstream. You can give blood to somebody who has AB blood. Okay, so your ability to donate blood you can only give to AB people who have AB, but you're kind of the universal receiver. You can get blood from anybody. So lucky you. If you are type O, it's quite the opposite. You can give blood to anybody with any type of blood, but you can only receive blood from those who are type O. Now, luckily, 43% of the population is type O blood. So for those of us that are type O blood, at least luckily we can receive blood from a good portion of the population, although not quite as universal. And then the Rh factor. So if you have the Rh factor, you'd be Rh positive, and you'd have the anti-Rh. Okay, so it's not exactly the antibody, but it's your anti-Rh. You can give blood to somebody who is Rh positive only, and you can get blood from anyone who is Rh positive or Rh negative. So it works the same way of you having the anti-Rh in this case, as opposed to it being an antibody, but it's the same idea. And so this is just a little bit more of the same sort of graphic. So let's kind of move along from that. Here's a little bit of those percentages I had talked about. So this breaks it down by ethnicity, but of course that can help narrow down too much. It's not something you want to use for any sort of stereotyping purposes, but it can help you narrow down the likelihood of it being a certain person's blood. Of course, then you can go to DNA, but blood typing is something you can do pretty easily right off the bat. So you can see that type O positive is pretty common, and it's actually the most common. The least common, and especially, let's see, I believe it's for Asians. Yep, here we go. AB negative, super uncommon for Asians to have AB negative. So if you have AB negative, it's just uncommon in general. Of course, the B negative is pretty uncommon for Asians as well, as is, I'm noticing a trend here, aren't you? That the Rh negative is really uncommon for Asians. So the Rh factor really comes into play there in terms of identification in that way. The Rh factor is something that came from, discovered from a rhesus monkey. Ooh, cute little guy. So 85% of the human population has a protein that is that Rh factor. And so if you have it, you're Rh positive. If you don't have it, you're Rh negative. And as we had talked about, the O negative being the universal donors and AB positive are universal recipients. So if you're AB positive, then you're going to be pretty lucky if you happen to be in that need. Any blood can be given to you. So if you're OH negative, you're probably a bit more on the short end of that stick, and that's why it's so important for there to be blood banks. So that way it's on hand for what you might need. And I think we've already kind of looked at this a little bit. Let's move right along. So here's some of those percentages we had talked about. So let's see, 43% is type O, 42 is type A, B is not nearly as common, and then AB is highly uncommon. So the questions come down to is it blood in the first place? Is it fake blood? And so those are fairly simple tests that can be taken. The alcohol content, is it human blood? Whose blood is it? And in fact, we can even take a quick look at that. So there's a few different tests that can be done on blood. So the Kastle-Meyer color test. So a mixture of phenolphthalein and hydrogen peroxide. So the hemoglobin will cause the blood to turn sort of a pink color. There's a hematest tablet. So it reacts with the iron group in the blood, so it causes a blue-green color. And there's a luminol test that will actually cause it to have a bit of a glow, and so that's something that can be sprayed on there, and a pretty easy identifier as to whether it's human or whether it's whether it's blood at all. Human versus animal blood. So we are technically animals, but non-human blood will typically have larger nuclei. So you can see this sort of like darker spot in the middle. That's the nuclei of the cell, and so you can't really see the nuclei with a reasonable microscope with human blood. So if you can see nuclei, it's probably some sort of non-human animal. So here's some other examples. You can see how the red blood cells look so very different within the various species of different animals. And then lastly, let's look at this precipitin test. So this is something that can help determine as to whether it's human blood. So what they will do is they inject human blood into a rabbit, and so the rabbit will produce the human antibodies, right? The anti-human antibodies, and then the antibodies are extracted, and then the anti-serum, those antibodies, will be put in with the human blood, and if it clumps, it is human blood because it is anti-human serum, essentially. And so, that's what the rabbit's producing. It's trying to produce a serum to fight off the human part, and it's when you extract it. From what I understand, this is not something that like harms the rabbit. It's not a lot that it's needed, and so that's a way to tell as to whether it is human blood or if it is animal blood. So that's a hopefully somewhat short one, and I think that will conclude our blood composition notes. And I just wanted to fill in some of those gaps. So I hope that's it, and you guys have a good one.